This is Esther Rakusin for WRFI Community Radio News. Last Saturday, September 9th, I had the opportunity to visit and experience the first BioBlitz to take place in the Cayuga Lake Basin. During the BioBlitz, many scientists and volunteers with knowledge of different kinds of organisms met to explore the property of Cayuga Nature Center and Smith Woods, observing, collecting, and counting samples of organisms of all kinds. I spoke to Alexandra Moore of the Paleontological Research Institute, one of the organizers of the event, about the idea behind the BioBlitz.
BioBlitz is a 24-hour activity where we get scientists from all sorts of different groups of animals and plants and every other living thing to come and try to identify as many species as they can in 24 hours. We're doing it because we really want to have a baseline for what the communities of plants and animals are here in the Cayuga Lake Basin. And we have two different venues or properties where this is happening today. Right now we're at the Cayuga Nature Center, and then just up the road there's also Smith Woods, right in the village of Trumansburg. And so we're going to compare what we find at the two places. Trumansburg, Smith Woods is an old-growth forest, and the ecosystem here has been much more disturbed by humans, so we expect to see two different things at the two different places. We have a mammal team, bird team, herpetology team, plant pathogen team, microbe team, aquatic organism team, fungus team, Lepidoptera, spiders. I think I got them. Lichens and mosses. Oh, I forgot the best one. And mollusks and annelids.
That's quite a range. And who is serving on the teams?
People from the community. Many of these scientists come from Cornell University, but there are also people from Ithaca College, from TC3, from Hobart William Smith. Many of them have brought their students, graduate students and undergraduates, and then anyone else in the community who has knowledge of some of these groups of organisms has been invited to come as well.
I see a group off to my left right now.
And so this is a public tour that we're looking at. And so in addition to the scientific endeavor, there's a whole program of outreach events all through the day. And so this is a plant walk being led by Dr. Marvin Pritz of Cornell University.
And what do you hope to do with the information that's gathered today?
I think from my perspective, the really important thing is community awareness. We want people who live in this area to know what's here and to know how special it is.
And why is it special?
That's a difficult question for a scientist to answer, because for me, anything in the natural environment is special. This is what we rely on as human communities. We need this ecosystem base that supports our human community, that helps us stay healthy. It's a great place for kids to come out and learn. It's just beautiful.
What do you think is your favorite thing about the area around the Cayuga Nature Center?
Well, that's a loaded question, because I'm a geologist, and so of course I'm really interested in the geology. We have an amazing suite of fossils that we find all over the Cayuga Lake Basin. We have really interesting structures and rocks that form our beautiful landscapes. This is why Ithaca is gorgeous.
So I know that mollusks are found in the fossils here, and so will the BioBlitz volunteers be looking at fossils as well as living things?
We really wanted to do that, but actually this property right here does not have very many fossils, so we passed on that and just stuck with the living things.
I next spoke to Scott LaGreca of the Cornell School of Integrative Plant Science, another organizer of the event. He talked about how he recruited the scientists and volunteers taking part in the BioBlitz.
Well, I'm one of the co-coordinators here. I was mostly in charge of getting all the scientists rounded up and out of their labs and into the woods here. We had to figure out organismal specialties, so plants and mammals and birds, all these taxon sort of specific groups, and figure out who would be a good team leader. A lot of times it's a professor, but not always, and basically getting in touch, making sure they can get out here on the day, and then getting a protocol down with them to survey, to to record the species. So I've run these sort of BioBlitz events before, and at the end of the day, people hand you all these like scratched, like lists of things with common names too, and so it's really hard to compile. So we insisted on like a digital iNaturalist.org platform where people can upload species observations, and that's how we're compiling the list. They've been very keen actually to come out here and look around and record. Yeah.
So I see from your name tag that you're an expert in lichens and mosses. And so I'm wondering, are you going to be participating as well, collecting information on those types of organisms?
Right, yeah. So my other role here is team leader for lichens and mosses, which are two very different organisms, but they grow together, so we consolidated them. Yeah, so we are compiling a list. We started last night.
What's your favorite lichen?
Oh my gosh. Oh, there's this fantastic lichen called the oyster lichen, which looks like little clamshells, and there's little powder that they produce, which are these asexual propagules. So that's how this thing gets around, and it's on pine trees, and we found it here today, which is cool.
The second floor of the Cayuga Nature Center Lodge was a beehive of activity as scientists and volunteers examined and counted various species. I spoke to Katie Rogan, a graduate of Hobart and William Smith Colleges, who was looking at various amphibian species.
I'm a recent alum of Hobart and William Smith Colleges, and an advisor of mine, Professor Brad Cosentino, started the herp team for the BioBlitz this year, and he asked if I would help out, and here I am. And I love my amphibians and reptiles and all that good stuff, so that's why I'm here. Last night we were out for a few hours setting some turtle traps to collect some turtles and tadpoles for this morning. And we walked along the trails and found some salamanders, some redbacks and newts and all that good stuff. So,
how do you set turtle traps?
So they're these big nets with metal rings, and you collapse them, and they have two sleeves on either end, and the turtles swim into them, and then they get stuck and then can't get out of them. We set little cans of sardines and fish.
And how did you find the salamanders?
So we go through the woods along the trails, a little off of them, and flip logs and different cover objects, and they're usually under there for us.
And what do you have here on display for the public?
So we have a small spring peeper that someone brought in for us. It's little and hops around a lot. We have a few dusky salamanders. They're small juveniles. And we have a green frog tadpole here, a little spotted salamander that just went through metamorphosis, so he doesn't have his gills anymore, also doesn't have his big yellow spots. And then we have a few newts who still have their gills as well.
So I see that some of the newts and the small salamanders are, well, about the same color as what you might find on the ground. So how did you find them?
So for these ones, we sample in streams. So it takes practice to train your eye to see all of them, especially with the toads too. They blend right in with your surroundings. So you have to be a little still sometimes and look for any movement that you can see.
And what is your favorite amphibian or reptile?
That's a hard question. I am very partial to redback salamanders because I've done research on them in the past, and they're just very abundant and great creatures. But I also like great tree frogs because you don't see them very often, and they're really cool.
Why do you think the public should care about preserving salamanders and tree frogs?
Well, herps and amphibians in general are just such a great indication of changes in our environment, and with what has been going on with climate change, they are just such a great indication of how we are affecting our ecosystem and how it'll continue to be affected by not only our changes, but everything else that's going on.
Next, I walked outside, where, under a large tent, two volunteers, John David Barrera and Carrie Dupree, were examining and sorting aquatic insects collected on site at the Cayuga Nature Center.
What we're doing is we're actually looking at aquatic insects as part of the inventory for the BioBlitz, and we're looking specifically at some of the streams that are in the area, as well as the pond that the Cayuga Nature Center possesses. And pretty much we're doing this as an inventory to figure out the diversity of aquatic insects in the area, and pretty much to help determine pretty much the quality of water and the quality of life that a lot of these insects and any of the organisms use, as part of, their daily life. So
Yeah, so what we're doing right now is preserving the insects in 70% ethanol, and we've taken kick net samples from the nearby stream. So by stirring up the sediment on the bottom of the stream, you can free up some of these macroinvertebrates, and then you capture them with a net. And then we also have been sampling in a pond. So we actually had one of my friends, Vanessa, she was in waist deep and stirring up the bottom of a nearby pond to get some of these organisms that we're sorting through right now. So we're basically just trying to sort them by genus, and then we'll take them inside to some experts who might be able to even sort them to species level.